Hey, so today in the first of 2 days, kind of in our short week, so we only have today Tuesday and tomorrow Wednesday that we're gonna have Zoom sessions this week. Otherwise, Thursday and Friday we have off. We're just gonna take a real slow introductory look at genetics, which is what we're going to be studying to finish up the first quarter and honestly first half of our year as far as content, if you believe it or not. But I just wanted to get started with this because I think that a lot of what we're going to look at today and tomorrow is going to be a review, so it won't add very much work on top of, reviewing and kind of studying for the cell processes test. But also, it'll allow me to see if some of these things that you looked at biology in ninth grade before are seeming a little bit foreign. It'll give me an opportunity to know that and make a plan about reviewing that stuff and not just going on ahead in any way. But today I just wanted to talk about like the basics of kind of Mendel's work and Mendelian genetics, okay, the things that you might have remembered doing, like the family traits project in ninth grade, or maybe even back in seventh grade when you had looked at some things with that. So I just wanted to go through that today so that we could preface kind of our work when we get back next week, when we go in a little bit more detail with some more complicated stuff. So the reason that I bring Gregory Mendel up is because he's basically the person who started modern genetics, or studying how traits are passed on from one individual to their progeny, to their offspring. And so Gregory Mendel was this really cool guy that he was a monk in the mid-1800s, and at that time, monks didn't have a lot to do. It was a very solitary life. They meditated and read scriptures and worked in abbeys that they lived, usually out in remote areas. And so he kind of had a lot of time for contemplation and for reflection and things like that. And during this time, he undertook some very arduous, very thorough experiments on pea plants, because pea plants were very easy to grow, and they had fast generation times. So he used the experimental method and quantitative analysis, which means collecting data, counting data, comparing ratios of the different types of peas that he grew, and through that very humble means of scientific experimentation, basically was able to discover the bases of many of the laws of genetics that we understand today. And so he's a very important figure to understand where our genetic understanding came from, but also as a perfect illustration of what can be done with a little bit of intuition, some creativity, and an understanding of the scientific method. So like I said, Mendel's work and his experiments that he undertook all were based on pea plants. And pea plants were really great to work on as a model organism for several reasons, especially for genetics. One, they had a bunch of different traits that were genetically heritable. So different colored flowers, so white versus purple, or color of peas, green versus yellow, or height of plant, tall versus short. There was a lot of these very easily observed discrete characteristics that were genetically based in pea plants. They were also really easy to pollinate. Get a brush or something, get some pollen from this one, pollinate this one. Pretty easy. And they were rapidly growing. So the time between the generations wasn't very long. So he could grow a whole bunch of them and follow generation after generation after generation, have a multitude of data in each generation, but then from generation to generation. So this picture kind of talks about the breeding of pea plants, which you do by cross-pollinating. Like I said, taking like a brush and rubbing it on the reproductive organs of one and getting some pollen off, and then rubbing it on the other and pollinating it artificially, kind of like that. So some terms that we want to understand when we're talking about his work, and one is called true breeding parents, and we represent them kind of with a capital P. So P stands for parents. And a true breeding parent is one which always produced offspring that looked like itself. Okay? So if I had a pea plant and I was interested in doing some experiments based on its white trait, its trait for white flower color. A true breeding white pea plant would seemingly always be producing in nature offspring that were always white. Okay? So that's kind of what true breeding means. It's an informal word. We're gonna talk about a more scientific word for that same term a little bit later, but they're always producing offspring that look like themselves. So P stands for parents. That's the first generation if we're starting from like generation 0, right? And their offspring are called F1, and F stands for filial, okay, F1. So that's the offspring of the P generation we call F1. And then if we pollinate F1 offspring, so maybe get some pollen from this flower and pollinate this flower with them, both in the F1 generation, and they would produce another generation, we call that the F2 generation. We could go to F3, F4, F5, okay, and things like that. So this is the basis of what he did. To true breeding parents, cross-pollinated them, created a whole bunch of offspring, F1 generation, picked 2 of the F1 generation plants, cross-pollinated them, created a whole bunch of the F2 generation, on and on and on and on and on, okay? There were 7 traits that Mendel collected data for, all at the same time. So think about having thousands of pea plants and looking at each one for these 7 traits. So flower color, the seed color, the seed shape, the pod color, axial or terminal flowers, tall or short plants, all of these things were examples of data that he collected about every single plant in every single generation, generation after generation. So you can start to get an idea about how time intensive and how energy intensive and how organized he had to be to get this correct. So this is kind of really briefly gets at the heart of what the data that he collected started to look like. So he started off pollinating true breeding purple flowered peas. So purple flowered peas that seemingly always produced offspring that was purple, and he cross-pollinated them with some other true breeding peas that had white flowers. And what he found is that in the F1 generation, so these plants, their offspring, were all purple. 100% of them, every single time. He repeated this experiment many times, and they were always all purple. So it wasn't just a coincidence. And he thought, where did the white flowers go? Why are none of them white? Well, I would have expected like maybe half of them to be white and half of them to be purple, or around that. But all of them were purple. Why is that? And so he took 2 of these purple flowers and cross-pollinated them, like we talked about before, to make the F2 generation. And he found that 75%, always around that same percentage, 75% from self-pollinating F1 flowers would be purple, and 25 would be white. And now the white is back. It skipped a generation, and now it's back. How could we self-pollinate a purple flower and get white flowers? And more interestingly, why was it, no matter which flower we picked, no matter how many times we did this experiment, it was always like 75-25, like a 3-to-1 weight ratio. It was never all purple, never all white, never 50-50. It was always this ratio. And so he looked at this and said, this isn't just a coincidence, there's something that's going on here. There's got to be some reason that I'm seeing these specific ratios. Every single time, it's always very similar. Why is this? And so we'll talk about Mendel's laws in next time, but one of the things that his findings meant was that traits came in alternative versions. So, for example, purple versus white flowers, those were different versions for the color trait, the flower color trait. And that these versions that Mendel called them of traits, later the name was changed to alleles. So an allele is a different version of a given trait or a given segment of DNA. So now we know, okay, because Mendel had no idea about DNA. DNA wasn't really a thing in the 18th. That would take until Watson and Crick before we really, understood the structure of DNA and all that. So he didn't know about DNA. He just, this is more abstract to him. Different alleles, versions of each trait, vary in the sequence of nucleotides. The DNA sequence at a specific locus or a location, locus also means location, of a gene. So here's a picture to help us understand. So if here are two homologous chromosomes, right? You have two pair of each chromosome. They come in pairs, 23 pairs. So let's say this is your first pair. You have 2 copies of your first pair. Here is a specific location. We call it a locus on the chromosome. The same spot in each chromosome. And at the same locus on each chromosome is where we find the DNA for a given trait. We find the 2 alleles, so allele 1 and allele 2. And so different alleles, he said, were varied in the sequence of DNA at this location. So this DNA could be different. So in terms of our flowers, there was a purple flower allele. There was a segment of DNA that would make a flower purple. And there was a white flower allele, or a segment of DNA which could make a flower white. So different versions of the gene at the same location of homologous chromosomes are different alleles. For each characteristic, an organism inherits 2 alleles, 1 from each parent. All right, this is very basic. All right, this is kind of Mendelian genetics according to Mendel's laws. When we talk about more modern genetics and non-Mendelian genetics, this can be expanded for where traits have more than 2 alleles and things like that. But for now, when we're trying to understand the basics, just a big disclaimer, this is true for Mendelian genetics. So in diploid organisms, so like ourselves, where we have or receive 1 copy of each chromosome from each parent, we have 2 sets of chromosomes, all right? We have like 2 editions of each chromosome, 2 sets of the genetic information from each chromosome, 1 set from each parent. What Mendel's findings mean, okay, and why he saw 100% purple in the F1 generation and then 75 and 25 in the F2 generation is because some traits are able to mask others or be expressed over the top of other traits. So purple and white flower colors are separate traits that don't blend. Okay? A lot of people at that time thought, like, oh, if you have a purple flower and you have a white flower, you'll make a light purple flower. That's not how the genetics work. Purple is able to mask, or we say dominate, the white color when both are present. And this brings us to our idea that's extremely crucial and fundamental for Mendelian genetics, is there is what's called a dominant allele. One of the 2 copies of the trait is dominant to the other. It's able to mask the other allele even if both are present. So it it is, for example, the purple color. So if on this picture of chromosome one of the alleles is purple, it doesn't matter what the other one is, this is always going to be expressed. Even this other one is white. It is dominant to the other one, right? And then we have examples. The other trait, the other allele is what's called recessive. And recessive alleles are only expressed if both of the copies are recessive. So for example, the only way a flower could be white is if both of these chromosomes would have a white allele. So you would need 2 copies of the white allele of the recessive allele for it to be expressed, for it to be visible, whereas you'd only need 1 copy of the dominant one, since it masks the other, to be visible. That's the whole idea of Mendel's discovery of dominance and recessiveness. That's the theory of dominance and recessiveness. Okay? Let's see. 2 other slides. Second to last, we came up with some terminology called genotype and phenotype. These words sound very similar and mean very similar things. Okay? A genotype versus a phenotype is the difference between how an organism looks and how its genetics are. So an organism's phenotype is a description of an organism's trait, the physical, what it looks like, what's expressed. So this flower has a phenotype of being purple, and this flower has a phenotype of being white. So phenotypes are descriptions. It appeals to the physical characteristics of a given trait. A genotype, on the other hand, describes an organism's genetic makeup, which alleles it possesses. So genotype has to do with genes, phenotype has to do with physical features. Right? These 2 words can get confusing because they're words that they're Their concepts that they define are closely related but separate. Okay? Lastly, the last thing I want to go through today is that we can represent alleles as letters. So for example, for the flower color, purple or white, we can represent the purple flower color with a capital P. So whatever the dominant allele is, the dominant trait, we represent that allele with a capital letter. And the white allele, whatever the recessive allele is, we refer to it with a lowercase letter. Okay? Now getting into what I talked about at the very beginning. What a true breeding plant is one that has both of their letters, both of their alleles that match. So a true breeding purple flower or purple pea plant would have 2 dominant alleles, 2 capital letter P's. And a true breeding white flower would have 2 recessive alleles, 2 lowercase p's. That's what true breeding means. 2 of the same alleles, okay? 2 of the same alleles. That's kind of where I want to stop today, because I know you have a test as well tomorrow, so I don't want to get too far into it, but I just wanted to give us a little intro to genetics. If this is a review, that's great, but if it's not, I am glad that I was able to go through this with you today.